Organic trace minerals come in many types and formulations, leading to confusion about chemistry, terminology, and methodologies. With Balchem's Quicheur line of chelated minerals, we provide superior performance and exceptional value by keeping it simple. Binding minerals to the highest quality plant protein-derived amino acids and peptides in our world-class production facilities, using a true chelation process pioneered by Balchem and trusted in both the human and animal arenas for nearly 60 years. The Quicheur line delivers proven and consistent bioavailability to maximize performance, and a no-frills pricing approach for greater profitability. Visit balchemanh.com to see how Quicheur chelated minerals are your link to superior performance and exceptional value.
Hello, everyone, and welcome to the Real Science Lecture Series. My name is Scott Sorrell, Director of Global Marketing for Balchem. Mycotoxins can produce huge economic and productivity losses in livestock, and in extreme cases, they can even lead to death. So what can we do about molds and mycotoxins in our feed? For today's Real Science webinar, we welcome Dr. Duarte Diaz from the University of Arizona. Dr. Diaz will be sharing some of the newest research concerning the interactions of fungal toxins, feed, and our animals. Dr. Diaz holds MS and PhD degrees in nutrition and toxicology from North Carolina State University. His research for the past 15 years has focused on the effects of mycotoxins in animal agriculture. Dr. Diaz has given over 40 invited presentations around the world and has published over 70 articles in scientific journals, proceedings, and popular press magazines. In 2005, Dr. Diaz served as editor of a publication focused on the applied impact of mycotoxins on agriculture titled The Mycotoxin Blue Book, published by Nottingham University Press. Welcome, Dr. Diaz. The floor is now yours.
Good morning, everybody, or at least very early morning for me. Not as early for some of you, but good morning anyways. I'll like to start off by thanking Balchem for the opportunity to be part of this series. Looking back at some of the previous talks and authors, I'm honored to be part of this group. I also take this time to be thankful to companies like Balchem for giving us, the extension people, an opportunity to broaden and expand our reach. We spend a lot of time with producers at a local level, and it's these types of interactions that give us the opportunity to not only share with a broader audience, but also learn from that broader audience. So again, I appreciate that opportunity. What I'll try to do today is it's a series of strategic storytelling, which I'm going to try to cover how I approach this discipline, which may not be traditional in the context of most other disciplines, or at least how I see it. And I'm going to sprinkle with some of my research over the years, or some of my collaborators' research over the years. But I'm really going to try to do I'm going to make an effort to do this, 30,000 feet and going in deep and then coming back out again to see the bigger picture. And again, I think, it's important to mention that as a student, I decided to specialize, as many of graduate students do this. But within my specialization, I also decided that I wanted to broaden my knowledge. So as I started as a nutritional toxicologist, over the years, my interests have broadened tremendously. And I do a lot of work in many of the areas that are related to these interactions between plants, molds, and animals, right? We do work in agronomy. We look at soil science, we look at meteorology, we venture into a lot of these other disciplines so that we are able to not only focus on our specific area of research, but give it a bigger meaning, look at the whole system. So that's what you'll probably get the most from my approach, is that although I'm really interested in understanding, toxicological pathways and how they impact the actual physiology or the actual biochemistry of the animal. I'm really more interested in how all these things come together to cause us the issues that we have to deal with. And again, I'll clarify that moles are not all bad. And I'll make some statements to the fact here in the next couple of slides. But I also will state that not toxins are difficult to describe in a conversation because people hear the word and automatically assume we're all going to die, right? Panic mode. And the toxins is within the dose, right? The dose is really what makes the effect. And we can live with fairly reasonable amounts of these toxins in our day-to-day, and you'll see that they've been naturally occurring for a very long time. It's when our systems start combining to multiple stressors that these concentrations or these lower concentrations may become more problematic. I, over the years, have stopped using definitions because I make the general assumption that a lot of people have heard mycotoxin talks before. That may be just or maybe unfair in some cases, but I'm going to go through these really briefly because these are some of what we consider the traditional definitions as far as the regulatory agencies or the institutions that monitor exposure to mycotoxins around the world. Mycotoxin is quite simply just a naturally occurring toxin produced by a fungi. The real important component of the mycotoxin is that it's a secondary metabolite of the mold, which in its essence is what really gives us a lot of the problems that we have with its proper understanding on when it's going to occur or not. So what that means is that the mold really has no defined reasons or biological, I shouldn't say that, some survival needs to produce these mycotoxins in most cases. A lot of times they're producing it as a result of some stressors. We'll talk about that in a little bit, but that makes it really unpredictable, meaning that the presence of mold doesn't necessarily mean that we're going to have toxins, and vice versa. When the mold can die and still leave this byproduct in a feedstuff, we'll find out also that the absence of mold doesn't necessarily mean there is an absence of mycotoxins. So we're going to try to roll along through here. Molds grow in a variety of feed crops. Again, we have our traditional commodities, and I like to generally describe feedstuffs within a range of risk. So a high-risk feedstuff, a low-risk feedstuffs. And again, there's also a regional component of it, depending on what region of the world or what climate in that specific region. We have a higher incidence or a higher tendency of certain mycotoxins to be contaminated. That doesn't mean that in the microenvironment, which is, a bin with feed stored in Alaska, you can't have the conditions that are desirable for, more tropical mold. And if that mold is there present, it's been dormant because the conditions are not favorable, and all of a sudden it changes, then it could actually flourish and cause problems. So we also have to be careful about how we, put on those regional, regional, regional standards or assumptions that we use in historically. Mycotoxins can cause a variety of adverse effects, especially to humans, and that is actually, as you'll see, one of the main drivers of a lot of our knowledge in mycotoxins. Obviously, if they're toxic in that context, they're going to be most likely toxic to human and livestock. And then again, we're going to try to talk a little bit of what differentiates, in our production system, sensitivity to these toxins. Obviously, we have some of these mycotoxins that have been described as carcinogenic. Again, that is the number one motivator of both research and regulatory control of mycotoxins, and we'll talk about it. But aflatoxin being, the grandfather of them. There is several agencies around the world that regulate exposure to mycotoxins, but mycotoxins are regulated for for exposure to humans and to animals, but it also often monitored by organizations, trade organizations, because they can also be used to control and exclude market opportunities. So it's really intricate how these toxins play a role in all these different areas of our production of food. There is actually obviously an international standard codes of practice that limit the exposure, and that's called the Codex. And again, we use that as a reference for many of the regulatory controls. That doesn't mean we have standardized controls. One of the main things you'll see consistently when you talk about mycotoxin exports and different markets is that, within regions, for example, US versus the European Union, there can be some very distinct differences in regulatory limits or acceptable limits for production. For the sake of the presentation today, I'm going to focus on this big picture, right? I'm going to really try to understand these biological factors that make crops susceptible, that make these plant mold interactions occur. And then I'm going to really look at, why those are consistently occurring in our system. Today, I'm not going to focus too much on the environmental factors. I'm just going to sprinkle some information on that. Again, we want to try to understand what is it within our practices that may potentially contribute to the overall contamination or increase the contamination, storage playing a very critical role. The rest is somewhere in the component on the lower part of this diagram, where we really want to focus on understanding the effects of these mycotoxins, toxicologically speaking, but also understand how they navigate through the food chain into the system. So again, diversion of stored material or harvested material so that the human population doesn't get exposed to this toxin generally means a higher concentration or more exposure to animal species, right? And then on animal component, if there are some potential carryover tissues or products, then that relates back to the human chain on this component. Fortunately for us, this is minimal, has been historically minimal. Aflatoxin in milk being probably the only one that has, major relevance or significance. But again, it's something that we still need to look at. Every time we have an emerging toxin, that's one of the first things we want to make sure that we're not having carryover through the production chain. Again, and for many of us in this conversation today, we really are interested in understanding, once we have this distribution and diversion and all these things occur, we're understanding really what is happening at the animal model, on the animal production component. Where are we losing profits? Where are we losing animal health? Where are we losing efficacy in our system? And that we're going to sprinkle some research on that too, so that we can get a better picture. But again, 30,000 foot, we're going to try to navigate a little further inside. Again, I tend to be really geeky or dorky about the subject. I don't have the same expectation of my audience generally. If you talk to any family member in my house, the word mycotoxin has been banned from the dinner table and many other similar conversations. But really, if you're curious about it, there is actually a significant amount of historical events that have been either directly identified as a mycotoxicosis or have been speculated through similar mechanisms throughout history. Here are two really interesting examples. One is a 16th century Dutch painting called The Beggars, where they're actually describing this ergotism that was common in the area in the time point. And primary source of that ergotism was the consumption of poor quality grains utilized for the making of the bread. The lower income families, or the poor families, were eating this poor quality material, and they were getting these gangrenous circulatory related issues and losing, extremities in the process. The other one is one that's more on the speculatory side, where there is some hypothesis that link the consumption of some of these ergots and the associated hallucinations and behavioral issues with these witch hunts in Salem, the Salem trials, right? Again, pretty drastic intervention from the group if it was in fact a result of eating some contaminated material. But it peaks up in a lot of history. There is actually a colleague in Italy that has spent a lot of time actually thumbing through historical events and trying to associate them with a mycotoxin. I used to make the joke that, you know, if you give a mycotoxicologist enough time, he'll find a link with any disease or any event to mycotoxins. And I'll explain why that is actually not that far off, because mechanistically, these mycotoxins affect very, very broad systems, and they could really be associated with a lot of different things. If you really want to geek out on this, I recommend you read this book. And it's really interesting anthropological study done by some researchers, I think out of Harvard or Yale, where they actually went to these communities that still practice some of the dietary habits and actually other habits from the 14th into the 18th centuries. And they actually looked through their consumption of their food and identified that these microfungi that were a significant contributor to fertility, high death, and often some behavioral issues during that time point. So again, there is some speculation and there's some hypotheses, but there has actually been some pretty interesting studies to broaden this. And again, if you really think about what's happening here, right, molds are ubiquitous in the environment. They are going to be affecting plant growth or maybe affecting plant life, interacting with plant life. And as we modernize and we start, cultivating these things in a more controlled environment, we are going to be exposed to these interactions that occur. So I think as long as we've been eating, seeds and these types of commodities, we've been exposed to these mycotoxins. I think the intensity of exposure is difficult to assess from those times because we can't go back and test them, and we didn't have the techniques, but it's likely that there was a significant contributor. I'm broad really slide here just to give you an idea of when we look at human disease and epidemiological research on the exposure of different mold and mold toxins. There's a really broad amount of diseases that have been historically linked, associated in actual epidemiological research with mycotoxin exposure. These are human diseases, I'll grant you, and again that drives a lot of the potential funding opportunities to do research in mycotoxins. So the list is broad. Again, and it can be really very specialized, very specific diseases like ergotism, which we just discussed, to things that are more broader in nature, like testicular cancer, and it's linked to ochratoxin A consumption in some populations, or esophageal cancer, for example, also in some regions of the world where corn consumption is a main staple in their diet. The one thing that I really want to make sure that we focus on is that even within circles of these people that are working on mycotoxin, you often hear this concept of sterilization of the environment or the feed, right? That we want to live in an environment where no toxins are present and no mycotoxins are going to be present, and that's really unrealistic and unnatural, right? The reality is that molds are, like I said before, ubiquitous. They're really important in the decomposition of organic material. They're very important in a lot of ecological pathways, and they're just a factor of these ecological systems. So we can't really expect to have a system where zero exposure to mold is expected and zero contamination of mycotoxin is the expectation. Again, what we can do is control the conditions through our manufacturing, harvest, and all these other practices to generate feeds that have manageable concentrations of these toxins that allow us to produce at a high level without impacting animal productivity, health, or any other area of our system. But we need to learn to coexist with this system, and it's something that we can't just eliminate from the ecology, right? Next two slides is really basic identification of the different molds that we see consistently in the feedstuff. One thing that's really interesting here is if you really do a deep search into the literature, you'll find that there is, hundreds, if not thousands of different compounds described in the literature. As far as mycotoxin, I think the current count is close to 500. But you'll notice that we don't, well, I mean, obviously by logic, we can't focus on all those toxins, but we tend to focus on a certain group of them. The group of toxins that we mostly focus are a combination of two really important components. One is their presence or their occurrence in traditional feedstuff, right? I mean, some moles are very consistently found in the commodities that we use for feeding animals. And as a result, their potential exposure to animals is much more critical of our knowledge, right? We require more understanding of it, even if that mold produced toxin in itself is not extremely toxic. It just occurs with very high frequency. A really good example of this maybe would be DON, where it's very commonly occurring, not necessarily the most toxic compound out there. Again, you can say something to that effect, but the other component of that is that we have toxins that are much less frequently found, but can be extremely toxic. They can be extremely damaging. They can be extremely impactful. Again, within the same category, T2 toxin is a very good example that in even small concentrations, they can lead to, very drastic effects in animal and even death, right? And that's the primary criteria why we focus on a very smaller window of toxin in our research component. You hear a lot about aflatoxin, ziranon, fumonisin, the T2 toxins. Like I said before, those are really the critical ones. But ochratoxin, citrinin, postulin are really important mycotoxin, especially in the human food chain. Alternaria toxins are also going to be really important, especially in the human food chain. There's other really common mycotoxins out there. There's other ones that are, again, really common and, not been described as potentially harmful. We all consume some way or another, moldy materials. Again, the bottom of this is a really good example of this moldy delicacy of Mexico. That is very common, right? I mean, this you'll see there in the left-hand side how it actually is seen in the corn, right? And then they'll take that off and cook it into your tacos or your tortilla in the morning, and it's actually considered a high delicacy. I am not very fond of it, so I will stay away from it. But it's really not because of any fear of potential toxin, because we have not been able to describe anything from that. But again, just wanted to give you a picture of how broad this discipline can be. Again, I get a lot of questions sometimes about these newly described toxins, and I'm like, well, again, I think until we have a better understanding and know how frequently they're occurring into the or if they're extremely toxic, they'll still probably maintain a lower priority on the arena of research and regulation because of those reasons. Again, any basic mycology course will tell you the traditional standards for creating a suitable substrate for mold growth. What I really want to focus here is that this is broad, right? I mean, we have molds that are adapted to cold weather. We have molds that are adapted to hot weather. We have molds that typically are adapted to different types of substrates. In general, some of the key targets are actually moisture or water related. Water activity, relative humidity, and moisture are some of the important triggers. Obviously, oxygen has to be present. But again, very adaptable species. I tell people a story all the time. In Chernobyl, after the nuclear plant, when they went back into that area, right, with their heavy suits and guarded for the radiation, the first thing that they found that was interesting was a mold species that actually quickly adapted to using radiation as a form of energy. And to this day, they're using that mold it's in the space station. They're using that mold as an example of highly adaptable species, right? So again, molds can really change their minds pretty quickly. Again, we joked a lot in our discipline that Molds must not be reading the same books that we do, because every time we think we have a understanding of their potential occurrence and pathways and mechanisms, they throw a curve, and we're back at the drawing board. But again, in our systems, we know what are those areas of potential risk. The field is one it's very critical. Our soils are heavily contaminated with fungi. They're part of that microbiota. Some of them very important for soil health. Again, those in the right environments can actually aerolize and expose the rest of the plant. Be exposed to the seeds that are our primary commodities and contaminated in that sense. After that, any place in our production system that has areas of stagnant feed or low circulation can be really hotbeds for mold growth and potential toxin production. So cleanliness, flow of feed, and movement of feed through the system is very important in control and reduction of mycotoxin exposure. But again, some of these are really difficult to manage in a traditional system, especially in a traditional high volume system. What I want to point out really quickly before we jump into any toxicology, that also you have to understand that as mold growth on feed, they're actually utilizing nutrients from that substrate. So it's not just critical to think about mycotoxin exposure. We have to think about lower nutritional value of that feed stuff in comparison. So again, I run an experiment with a mycotoxin contaminated feed and a clean feed, although that doesn't really exist, but less contaminated feed, and I see differences in performance. I automatically make the assumption that those are effects associated with the toxin. I run some nutritional value on the two feedstuffs, and then I realize that they're different in their nutritional value, right? The presence of that mold, the growth of that mold had a negative pathological impact on the quality of that grain or that material that you're talking about. So again, this has been described, some work out of Brazil, but just really basic study where materials were stored at 12% or 15% using that 13% humidity as that cutoff. We know there's molds that can grow at lower percent humidity, but that's the traditional one for the ones that we commonly speak of. And the AF in that is antifungal component. And basically what this experiment shows that at 15, you're starting getting losses in energy, right? Fat content is significantly reduced, and you get significant reductions in specific weight. So those two feedstuffs are not the same. They're not going to feed the same. Animals are not going to perform the same. And again, you can guarantee that effect is mostly associated with that mold growth, because if you look at 15% with the use of the fungicide, you prevent that. Again, the same similar effect with the specific weight, right? Effects in protein here were not visible, but there is some specific literature that shows some amino acid changes in moldy or mold infested materials. So again, keep that in mind, that when we're talking about moldy feed, feeding of moldy feeds in a production system, we're also talking about the loss of nutritive value in that feed stuff as a consequence of that mold growing in there and utilizing those materials. But what we really came here for was to really talk about what happens when those molds decide to bring with them some friends and create a little bit more havoc. Frankly speaking, we really don't truly understand why molds produce mycotoxins. We have hypotheses, we have theories, we have specific descriptions of a specific mold and toxin effect, but in a general sense, we can't really go out there and say molds produce toxins because of X, Y, and Z. The three primary working hypotheses, and the ones that have been associated with the greatest number of mycotoxins, is one, that molds are producing these toxins as a response to stress. We see this all the time, even in process where we're actually physically trying to kill the mold, like the utilization of antifungal material, propionic acid, or something like that. As the mold is dying, it's producing some mycotoxins in that process. Again, that might seem counterproductive, but the reality is that you really want to kill that mold and maybe potentially release some mycotoxins, as opposed to leaving that mold there being exposed to changes in environmental conditions and continuously producing, toxins. But we see that all the time. It's pretty common occurrence. The second working hypothesis is that most produce these mycotoxins as a competitive advantage in this substrate war, right? I mean, let's look at this, a feed stuff as a micro ecosystem. And in that micro ecosystem, there's all these microorganisms competing for dominance, right? And if I am a microorganism in that system and I can produce a molecule, a compound that is toxic to some of my competitors, then that gives me a competitive advantage, right? It gives me that dominance in that substrate. The reason why we're really keen on this hypothesis is that many of the mycotoxins are also very potent antimicrobial. So again, classic example of this is the historical example of a mycotoxin, which is penicillin, right? It's a fungal produced toxin that is, very antimicrobial. And again, we see this with a lot of the other mycotoxins that we have in the in our system, in our research base. And again, as we get more savvy in things like the microbiome, we'll start getting a little bit more information about how those potentially are affecting. In the ruminant side, we've been talking about that for a while. We believe that, in certain conditions, mycotoxins in the rumen are having actual effects on populations of the rumen and potentially causing, problems and by that mechanism. The last mechanism is one that's more specific. Like I said before, sometimes we find ones that are specific to a different mold and a toxin. Here's an example of Fusarium graminearum, and you'll see on the right, this is a research study where it actually compares 4 different strains of Fusarium graminearum. One that does not produce DON, deoxynivalenol vomit toxin, however you want to call it, and the 3 other ones that do a different aggressiveness, let's say. And what they did in this experiment is that you go in there and you poke one of the seed heads, right, and you inoculate it with the mold, and you wait 18 days for this example, but it's really about a week, and you go back and you see how much growth is of that mold and how much is spread throughout that spigot, that seed head, right? And basically what you'll see is that with a non-DON producing Fusarium, when you come back 8-10 days or 18 days later, what you'll see is a lot of mold growth in that seed, but none in the adjacent seeds. When you put a DON producing strain, again, as you'll see in those top 3 curves, what you end up seeing is that mold produces actually DON to cause micro abrasions, irritate the covering of the adjacent seed, and then allowing it to propagate throughout that entire material. So pretty neat mechanism. But again, at the same time, if we look at these trichotism mycotoxins and we look at what the effects we often see in animal species, we'll see that that's, again, irritation of dermal tissue, irritation of intestinal tissue are commonly associated with them, right? So no, the mold did not produce that to irritate animals. It probably produced it to propagate throughout that seed, but it's a consequence of that, evolutionary trait, or that, that we have the issues associated with intestinal irritation in a lot of animals exposed to, especially Fusarium trichotisins. So I'm not going to dwell too much on this. I really want to make sure that you guys truly understand that because of the ubiquitous nature of the molds and because of the many factors that potentially affect mold growth, mold infestation into a substrate, and potential toxin growth, we can have mycotoxin occurring at any point in our production food chain, right? I mean, I tell people the example, right? We have this phenomenon in the medical field that's called sick building syndrome, right? That in wards for immunocompromised patients, they have to scrub walls, continuously to sanitize, because molds will grow. They'll find substrate in pretty much anywhere, and those molds can, cause havoc on these patients that have immune system that does not properly function. At the same level, once you prepare a finished feed and you throw it in the bunk and have your animals eating it, right there in that time period where that material is being exposed to consumption through the animal, there's enough time for a mold to grow, propagate, and actually produce toxins in there and cause issues. So we spend a lot of time trying to find blame and causation in a lot of the issues, but the most likely events is that you're getting some mold growth in the field, some toxin production in the field. You're getting some during the harvest and storage processes, and then you're also getting some during that. And that consequence of that is all those sets of conditions are different. What you end up getting is a combination of different mycotoxins, all of them produced at different stages of that full cycle when conditions were more favorable for that specific mold to grow. And again, that's pretty well described in the literature. Typically, when you have one mold, you will have more than one mold, right? It's pretty classically demonstrated in the literature, but it also causes a really big problem for us scientifically when we try to really understand how overall mycotoxins have an impact on production, because we can do research in many ways, but traditionally we will take individual toxins and put them and expose them to our animal model and extrapolate information from that, when in reality we don't understand if the combination of these molds affecting different mechanisms, different systems, is really much more critical than that. Some people, us included, have historically done research on multiple contaminated feedstuff, but at the same end, when we go to the other side of the scientific community, there's a lot of criticism because we don't really know which one of those toxins was the primary toxin causing the primary effect or the effects that we are associated with. So it's a really difficult problem to have to deal with consistently when you're doing mycotoxin research. You add that to that component that you can actually utilize synthetic or chemical grade produce mycotoxins, or you can actually use culture material. And the literature clearly demonstrates that those have differing toxicity, right? So again, a clear, perfect mycotoxin research experiment is probably impossible in our current knowledge, right? I'm going to jump really quick to the analysis, because I think it informs a lot of what we know right now on mycotoxins. If you go 50 years back, you realize that we have the capacity to maybe test for one or two mycotoxin in pretty robust methodologies that took a lot of time and a lot of chemical use. And you jump right now to our current methodology where we have this LCMS MS equipment that will do 25, 30 associated mycotoxins in one run. We have the capacity to detect mycotoxins up to parts per trillion, et cetera, et cetera. We've definitely gone significantly better at mycotoxin analysis. This is both a good and a bad thing. In my general idea, I think sensitivity is a double-edged sword. As we become more capable of finding these toxins in lower concentrations, at the same time, regulatory agencies will likely look at these numbers and continues to reduce that, independent of what the toxicological information informs us. I think that's a double-edged sword, but the sensitivity also provides us another potential tool, which is being able to actually identify residues of that compound in the animal and use that as a model for research. And I'll explain that because why that's important, because in general terms, the feed can be very difficult to analyze because of some issues associated with sample collection. But currently we have a little bit of everything, rapid monitoring that you can do right there immediately in the system, semi-quantitative. This TLC that you see there is still very commonly used around the world. It's probably one of the most common methods. And then you have the HPLC, which is our historical standard. Now with these LCMS methods, we've gotten really far in that. In general terms, I think the real big problem we have with analytical procedure is that often we forget that the analytical procedure is not what you do in the lab. It starts at the collection of the sample. And this is critical because if you have a lot and you collect a sample from that, you're having the expectation that 2 kilograms, 1 kilogram sample is going to be representative of the entire lot. If you have a compound inside of that lot that is homogeneously distributed, this is not an issue. But with molds, what you'll have is they'll grow in specific areas on that lot where conditions are favorable, oxygen exposure, water exposure. And as a result, if you take a sample from the wrong part of that lot, you're going to misrepresent the analytical procedure. Again, every one of these arrows in black here would represent what I would call a potential analytical error. And depending on the method and what you're analyzing, one of these arrows or two of these arrows would be more important than the other. I'll try to describe that, how that works in the mycotoxin. But Dr. Whitaker at ARS, USDA, down in Raleigh, spent a significant amount of his career building mathematical or statistical models to better understand this distribution, right? And what he actually found was that if you look at this, you here on the left, you have the sample, a subsample, the actual assay, and the total error variance and of the analytical procedure, right? And what he actually found was that for at least aflatoxin, 75% of the error is in the collection of the sample. And there's another 16% of the error associated in the subsample. So let's say you got 1 kilogram and you reduce to this 50 grams, which is what you're going to analyze. You put another 16% error into that system, right? So often we spend inordinate amount of time discussing of what analytical method is better, when in reality, we should be focusing a lot more on how to collect samples and reduce the total error associated with the sample collection. I'll give you a really brief example. This is a lot of grain, 2 samples from each, just to look at protein. If you take this one and you take that one, yeah, you're going to be really close to the mean protein concentration of that, which is the sum, it's calculated from all these individual samples. With aflatoxin, you take the same 2 samples, or you take that same sample used for protein, you're going to get a 0 on this one. If by chance you took this one and that one, now you're going to get 100 parts per billion. Both of those are incorrect, right? You have buyers and seller errors introduced in here. Only 10 parts per billion is the total concentration of all the entire lot, right? So you have to be really critical on this, meaning that if you're going to spend the time and effort in actually knowing what mycotoxin contamination is in the system, you can't use our traditional assays or our traditional protocols for collection of nutrient-related samples. You have to have something specialized for mycotoxin that allows you more robustness in that. Again, really quick example from Dr. Whitaker. He really just here shows you that if you increase the sample size from 1 kilogram to about 5 kilograms, you are significantly reducing about 80% reduction of the sample variance, right? So the larger sample, and again, when I say larger sample size, I don't mean just more feed. I just mean more subsamples to mix into one big sample, to mix into one final sample, right? But just by doing that, again, setting up a protocol specifically to address the variance in the mycotoxin assay, you can significantly reduce the amount of error associated with that. Then we can start having discussion about whether HPLC is better than ELISA, et cetera, et cetera. The other thing that's really important is we need to constantly remove our visual biases from the system, right? We go in there and select feed from the moldy part of the feed when we want to find mycotoxin, and when we don't want to find mycotoxin, we look at the really clean one. Here's a really basic example. You look at left to right, really moldy, really ugly looking corn, and the really traditional normal size, normal looking corn has the incredibly high concentrations of 263 parts per billion and parts per million of DON and 83 parts per million of zearalenone. This one kernel in a ton of feed is enough to cost you a significant amount of losses in a swine operation, for example. A big question. Oh, sorry. Ooh, I don't know if I can remove that. Hopefully you guys can see that. A big question is in the traditional sense is there more mycotoxins now than we had before? And reality is that there are some reasons to suspect that we've created some conditions that are increasing mycotoxin contamination. We obviously find more mycotoxins now because we test for more mycotoxins, but there are some reasons for us to believe that there is. Some of them are related to the environmental conditions. Others are related to how we actually grow crops and how we select hybrids for production. And then the other side of that is that we may also be addressing in the same conversation sensitivity issues related to our animal models. Most of those really related to an increase in animal production or intensity. And I'll talk about those really briefly. I'm not gonna, dwell on the meteorology side, although I think it's really fascinating. But if you look at this in the bottom one specifically, you look at the frequency of El Nino and La Nina, and you start looking that once we start passing these 1980 threshold, we start seeing El Nino, La Nina occur with much more higher frequency. If you live in the United States, you know that El Nino has a drastic impact on climatological phenomena in the United States. And those are transferred into susceptibility of the plant for mold growth and potential mycotoxin contamination. And those are pretty well described in the literature. The other side of this is based on the agronomic that I spoke really briefly. I mean, if you really think about, some of the common practices, this is really looking at the impact of tilling. Again, there's a good reason why we want to go to no-till or limited till, related to the fact that the nutrients and the nourishment of the soil. But the reality is that the molds growing into that system also have an impact when there is no till. They're more viable, they're more consistent. So if you have a field that has a high level of fusarium growth from year to year, it may be very recommended to actually go in there and do a till and actually expose those molds to the environment and reduce that cycle. In this basic example, basically what it shows is that at a no-till site, there was much higher concentrations of DON compared to the heavy till side, and it was pretty linear depending on the intensity of the till. Again, not saying we should go back to full tilling. I'm just saying no tilling has some benefits, many benefits, but one of the potential drawbacks is that in a system like this, where you have this intensity, you may need to periodically think about tilling and breaking those mold cycles in there, or the dominant cycle of that mold in that system. I talked a little bit about the animal side. I think animals are under generally more production stress. I'm not saying the animals are stressed. I'm saying that the system itself has a much higher expectation. So again, associated with a car running on the red line or very close to the red line, if you're behind that red line, you're performing at a very high level. You just don't have a lot of room to fluctuate on that other side of the red line, and that's where we often find ourselves very close to that system. Maximizing that system puts you in that space. I think there's a potential, and there's some literature about the genetic vulnerability. Again, turkey is a really good example. I don't have time to go too much details in there, but we have seen that throughout the history of domesticating this animal and increasing its capacity and production, they've lost some of their mechanisms associated with detoxification of aflatoxin, for example. So I think those are pretty easy to understand. I think we see those and we talk about those in the production system. I'm going to finish off the presentation here in the next couple of minutes, just basically talking about the toxicology of these mycotoxins in the context of what I think is important to us. So don't concern yourself if it sounds a little too technical, because that's really not the scope. And then I'm going to sprinkle it with some of the research and how we think about research in the component and how that's related with the specific mycotoxin systems, right? Aflatoxin is really the grandfather of this discipline, where it really all started, at least from the animal human side. Disease side, again, aflatoxin is an incredibly powerful molecule. It's been described as this most potent naturally occurring carcinogen. It's absorbed pretty easily in the animal system. It is converted to this epoxide, which is the active compound, and as you'll see here, that epoxide will bind to the DNA molecule, causing replication errors that lead to mutation and cancer. And that's really at the core of a lot of our concerns and research on aflatoxin, especially as it relates to human and our terminal animal production species, right? We're not typically growing animals that long that we get a chance to see those effects, but that same epoxide has a really important protein binding effect that leads to hepatotoxicity. So aflatoxin is apart from being carcinogenic, is an extremely strong hepatotoxin, and it's been linked in humans to correlated associated effects with populations with, for example, high hepatitis B, and that combination, it can be really critical. Again, it's not uncommon to see it in the literature that straight aflatoxin exposure in human populations can lead to death and mortality. Obviously, if the population has other underlying medical issues, that increases. But it's in the literature. We see it. Again, the levels are high for those to occur, but it happens. In our models, what we really have to focus is how aflatoxin interacts with hepatic health in a lot of production. So we look at our production models and our high intensity production models. Aflatoxin is often associated with those periods of high stress in the liver. Again, a really good example for me, obviously coming from a little more of a dairy background, is that early lactation, when the cow is mobilizing a lot of body tissue and is utilizing the liver to compensate that, to process that, right? And the reality is that the inclusion of hepatotoxin in that time point, where the liver is critically functioning at 2x, 2x, 3x, it's a very powerful combination that can lead to a lot of problems. As a result of this issue associated with aflatoxin carrying on into the milk, we spent a lot of time looking at this. Again, I'll show you in the next slide some of my master's research, but in my master's, we basically developed a model to use actual full marker, right, aflatoxin M1 of exposure to understand if a binder or a sequestering agent was actually helpful in that situation. But over the years, we looked at things about is the permeability of the mammary gland affect passage, right? Are animals often passing 3 or 4% aflatoxin? Others are concentrated more in the 1 to 1.7, 1 to 2, which is the more traditional range. And we found that the permeability of the mammary gland doesn't affect, high somatic cell counts are not, cows are not producing more, but we see some pretty interesting species to species differences. Mammalian differences in the carryover of aflatoxin in milk. We've also looked at where we put the material and how it can potentially have an impact, and we found that if you mix the sequestering agent or the material in a place where it has the more time to be exposed to get distributed in the feedstuff, because most of that sequestration is going to occur at the GI tract, you increase the capacity or the potential of that product. And that, again, it's pretty interesting, but also leads to a couple of headaches because they can manipulate the research model. Again, here's my master's research, really just designing this model has been really powerful to be able to test if a sequestering agent is actually working. The other thing that was really interesting is that we compared in this in vitro assay with an in vivo assay. In vitro would be really the ideal way for you to test a product and say, hey, this has a good potential, or no, it doesn't have good potential. And what we found was that the in vitro assay was a very poor estimator of in vivo efficacy. We had products that would bind 99% in vitro and then would not work in the system with all the complexity of the GI tract of these animals. So again, that was very important discovery, but more importantly, this model of actually testing in animal has been really powerful over the years. Again, if you look in this model, also one of the things that we were looking at is regulatory restraints. Right, in the United States, you can't feed more than 20 parts per billion because the accumulation of 20 parts per billion using a transfer rate of about 1.7 leads you to potentially be close to the risk of contamination. In our case, we went in and tested the with and without, and were we able to prevent, in this specific case, animals from going above the regulatory limit, meaning that, for a marginal investment in an operation, using this product as a preventive could have been the result, or could have been the difference between you having to dump milk or not. Now, having said that, that doesn't mean that any dose this is going to happen. This is dose dependent. If we would have increased the dose of aflatoxin, we might have even exceeded that in the treatment group. Again, it's about 1.7. Normally, we reduce that to about 50% with the use of these compounds. A really interesting mycotoxin is zearalenone. I think I'm getting close to the end here, so I know we're close to the time, so I'm gonna zoom through a little bit here. But zearalenone is really interesting because toxicologically speaking, it doesn't really cause a lot of issues. The molecule itself is not that aggressive, but it actually functions like any phytoestrogen or environmental estrogen, which is it just mimics estrogen and it binds to the estrogen receptor. And that response is going to be really critical depending on the amounts of estrogen receptors and the type of estrogen receptors in animal species. And that's why we see a really broad difference in effects with zearalenone in different species. Again, human exposure to these compounds has been described also in the literature. It's been associated with telargy. Again, hits a little bit close to home. I'm originally from Puerto Rico, and one of the places where this was at least hypothesized in the literature that there had been a exposure to a estrogenic-like compound in some food that led to this over early development of young females, right? In our sense, we've really interested in really understanding why these two really common events occur in our system, like the swelling of the vulva and these, swelling of the mammary gland in dairy cows are both really emotional things, right? When producers see this thing, they panic, they run around. But in general terms, when you look at the molecule of zearalenone, it could be a growth promotant, right? I mean, Zeranol, which is a derivative of zearalenone, was actually marketed as a growth promotant. So we often don't know if these symptoms or these things are associated with negative impacts. But again, we spent a little bit more time looking at this, and we saw that yes, the zearalenone associated effect is having significantly impacts in the gland development for these animals, and they potentially will be less productive in the future. Femininism is an interesting mycotoxin. Again, when I was talking to you guys about having newer mycotoxins and them entering into the mix, Femininism came in, took everybody by storm, and then all of a sudden changed a lot of what we thought about mycotoxins because of its important impact in human esophageal cancer primarily, but other cancers. But also has been associated with interfering with development in pregnant women. And one of the primary mechanisms is described through this sphingosine mechanism, but it also apparently interferes with folate uptake. So that's been associated with these growth and development issues in children, but also this sphingolipid inhibition is very important to a lot of the primary mechanisms in our production system. And we really think about the where we see a lot of the problems is we see pulmonary edema in pigs. We see encephalomalacia in horses. We see more kidney-related issues in other production species, primarily in ruminants. So again, same toxin, very different described mechanisms. Again, I think it's mostly associated with the intensity of either blood flow to specific organs because of the needs, or some prior established prioritization into the system. But interesting enough, right, we can see the same toxin have really drastically different effects in species as that moves along. And I'll finish off here with DON. DON is with trichotisins. Trichotisins are probably, interestingly enough, one of the least described methodologically in the toxicological side. We have a lot of really small pieces of the puzzle. Nobody's really put the puzzle really greatly together. There's a lot of really good work done out of Guelph with Dr. Trevor Smith and Dr. Halati Swami. And they did a really interesting work. And one of the things that they came out of that lab was that they understood that trichotisins had an effect on this amino acid catabolism. And as a result of that, you started getting a increase in tryptophan in the blood flow. At the same time, some of the associated mycotoxins could be binding albumin, so you have the elevated tryptophan levels in the blood, less albumin to bind it, more free tryptophan, and that free tryptophan crosses the blood-brain barrier and reduces, appetite intake and causes lethargy in the animals. And again, so much so that the DON is actually patented as an appetite suppressant in the patent world out there. I would not recommend that, by the way, because it also causes irritation and immunosuppression. But again, pathway wise, that's one of the primary reasons why pigs go off feed. We often associate it with just a toxicological response. I don't like that. It's making me feel bad. I don't want to eat it. But in reality, it's more neurological response. But like I said before, T2 toxin is also it's in the same category. It's pretty intense and can be causing very drastic irritation of the intestinal lining. Again, typically leading to a secondary problem in the animal, and your diagnostic of that T2 exposure becoming much more complex because what you're seeing is a secondary response. But if you look in the literature, there's really well described papers that look at how these specific toxins are irritating intestinal tissue from the sense of production. Again, taking that 30,000 foot back, I'll look back again. We irritate that intestinal tissue. We increase the maintenance energy. We're losing energy for production, and that's typically what we see, right? This spigot open, wasting water and making it really difficult for us to be profitable in the system. So I'm going to jump from that to one last slide. I think having moved to Arizona made me also have to really start thinking about the system in itself. And one of the things that I thought was really interesting is if you take a traditional heat stress image, right, of the effects of heat stress, and you wanted to take these little stickers and say, does this look like something a mycotoxin could also affect? It's almost 100% of all the symptoms or all the associated effects with heat stress could be exacerbated by exposure to mycotoxin. Obviously, the classic one is ergots, right? We all heard the stories about the cattle, in the lake, cooling off because they've had some ergots. But again, you start looking at things like reduced feed intake. We just talked about the effect of DON on intake. We looked at immunosuppression and how potentially you also have lower immunity, oxidative stress associated immunity in the cattle. We talked about intestinal health, intestinal irritation, the same thing during heat stress. So right now we're trying to build models where we can actually do low level toxin exposure. This is one of the big limitations of a lot of our research. We have to use really high concentrations of mycotoxins and heat exposure to build a model that really mimics what we may be potentially seeing in the field. And with that, I'd like to say thank you again to the sponsors, Balchem, for the opportunity, you guys for taking time of your day watching, whether you're watching it now live or potentially later in a recording. And I'll give you access to two references that I worked on that I'm fairly proud of. The Blue Book, as you heard before, is a pretty general reference book. We really wanted to look at the applied aspects of it in this publication. Probably needs an update. And then, I've been writing this feed stuff references issue about mycotoxins the last, 10 years, some of them with Dr. Willow and Dr. Hagler. And but I don't know if feed stuff is actually doing this anymore in the future. I haven't heard from them this year. But again, that's also a really good updated. I update it every year, reference on all things mycotoxin. And with that, I would really like to thank you guys again, and I'll open it up for questions.
We did have a couple of questions come in, and I know we are over time, but I will ask you one question here from Jamie. Jamie's asking, what's the best way to dispose of moldy silages on farm? Is spreading the silage back on the fields contributing to mycotoxin occurrence?
Potentially. I think, again, you have to have a better understanding of, what molds are in there and what their survival mechanisms may be. A lot of times spreading them in the field exposes them to the sun intensity and the temperature. And that's a really good way of inoculating, of killing them. But you really do have to be careful if that's a field that you're going to replant with something similar. That might not be a very good pathway. Again, I applaud anybody that throws away moldy feed, because I unfortunately see a lot of people trying to feed it more than get rid of it. But again, if you can put it in a field that is not necessarily a field that's going to grow that same crop, that would be excellent. That would be a lot better than putting it in any other field. Or if you can just spread it out into a compost pile or something like that, that might also be a viable way. If the mold is still present, if the mold's not present, just having the toxin there on the ground is not really going to recontaminate anything.
Wonderful. And I know we've passed the top of the hour by a bit here. So just as a reminder, we will have all of the questions that we're not able to answer this morning. We'll send them to you so you can answer them for us online. We'll give you one more question here. If a monogastric ate mold without mycotoxin present, can the animal still have adverse effects?
Yeah, I mean, in the literature, there's well-described mycosis, right? I mean, I think, aspergillosis is a common one that we've seen historically. A lot of times it's mostly associated with some respiratory issue. So if the feed is moldy and the animal's consuming it and it's aerolized and he inhales it, what does that mean? If he inhales it, then you're gonna potentially expose the lungs of that animal with that mycotoxin. But yeah, mold growth, and fungal growth and yeast growths are often common occurrence. If that GI tract is healthy, it'll probably be able to cope with a lot of that. If that GI tract is unhealthy and is having an issue, then you may be potentially contributing through the eating of the moldy feed. We eat a lot of moldy stuff. Not every mold is problematic, but again, that's the risk management there. You're taking a gamble in each direction by making that decision, right? If you have the opportunity to reduce risk, then you probably take the opposite solution, which is what we just recently discussed, which is finding a way of disposing it. Obviously, diversion is very important in mycotoxin management, meaning utilizing of the higher risk commodities or the higher risk ingredients in animals that are more resistance either because it's a different species and it has more resistance, or more resistance because the level of production doesn't put them in these sensitive time periods. So there's a lot of different things that you can do. I always tell people, right? I mean, you open the bread, it's got mold on it. There's really two types of people: those that throw the whole bread away, those that try to take the moldy out. Again, if you've ever seen any documentary that shows how far that mold is spread, even though you're only seeing a little batch, you'll probably be throwing out the whole bread. But if you grew in a home like mine, you ate a piece of bread, a sandwich with a couple holes on it. But I think the when in doubt, throw it out is a really smart way of thinking about it for a person like me that sits behind a desk and doesn't have to make monetary decisions. I think when you're in the real world and you actually have feed that you have to make decisions on, that you've paid money on, that you have to account for, then you start looking into alternative strategies, right? Should we feed it? Should we feed it to more resistant animals? Should we sell it to the neighbor that we don't like? Etc. I'm kidding. Do not sell it to the neighbor that you don't like. Right. But in general terms, it's complicated. I guess it's a good extension answer. It's complicated, but yes, animals can be exposed to those molds through consumption, both through aerolizing and consumption, and also damaging and colonizing some intestinal environment.
Well, thank you so much, Dr. Diaz, and thank you to everybody for attending today's webinar. If you do have additional questions, please submit them to anh.marketing@balchem.com, and we will submit them to you and have them up on the website going forward. The Real Science Lecture series of webinars continues next week on April 19th, when Dr. Eric Chapio talks about the interactions between nutrition and animal cognition, how diets can help to make animals smarter. And then on Tuesday, May 3rd, Mary Beth Hall from the USDA will discuss manure evaluation for reading your cows. Visit balchem.com/realscience for more details and to register. Balchem's podcast series continues to offer a deeper dive into our webinar topics. Log on to your favorite podcast platform or visit balchem.com/podcast. Subscribe to the Real Science Exchange and send us a screenshot along with your address, and we'll send you a cool Real Science Exchange t-shirt. On behalf of Balchem and Dr. Diaz, thank you for joining us today.
Thank you.